In this tutorial, we will discuss the Interactive Exploration module. The objective of this module is to allow you to design figures and quantifications that will be most interesting for analyzing your data. To this end, ShIVA offers you three types of representations. Firstly, two-dimensional representations such as map representations, like PCA, tsne or UMAP, as well as scatter plot representations. Secondly, one-dimensional representations in which we find violin plots, histograms and density curves. Finally, table representations to represent your quantitative data in table form. Let's start with the two-dimensional representation. To define the representation, you have access to several tabs which allow you to establish the figure as you wish to represent it. The first tab concerns the definition of the axes. It allows you to choose the x and y axis of your representation in the form of either a previously determined map, for example the PCA or UMAP that has been previously calculated, or in the form of metadata such as the percentage of mitochondrial or ribosomal genes. Or in the form of a feature, that is, gene expression. By combining these options, you can have very varied representations. If we choose maps, we can choose the UMAP representation on the x and y axis and perform a first plotting. Once the result is displayed, we see the UMAP representation that was previously calculated. Thanks to the second tab, we can then associate a color to each of the cells, the colors being provided by one of the metadata or features that are available. We find, for example, a clustering previously calculated with the Leiden method. We choose this clustering and ask for the plot. We observe that the UMAP representation now shows the different clusters. Note that these displays, unlike the previous ones, are dynamic and that you can use your mouse to get additional information for each of the indicated points. You can also activate or deactivate some of the annotations. For example, here by clicking on the 1, we deactivate the visualization of cluster 1, and by clicking on it again we reactivate this visualization. This allows us to eliminate some information from the figure to better see others. We can also choose to represent the data according to the classification provided by the HTO demultiplexing analysis, which allows us to see the original sample. We then request for the plot. Here we see that the colors have changed and now represent the original samples. Here it is extremely interesting to eliminate some of the samples from the representation to better visualize how the cells are distributed. It is also possible to use the Layout tab to request a different display using Facet mode. The Facet mode allows to separate the initial plot into several plots according to one or two chosen metadata. If we choose, for example, the classification by sample, instead of one plot, we will have 4 plots, one for each original sample. This makes it easier to identify the differences between each sample in the distribution of cells. If we now return to the Color tab, we can now choose the feature option which allows us to select a gene. Here we will choose the CD3E gene, and request its display. The color represents the level of expression of the gene. The more orange-red the color, the more the gene is expressed, and, as the layout we applied earlier is still active, we can see the differences in expression of this gene across the different samples. The Theme tab allows you to choose the graphical representation. Several themes are available. By default, the classic mode is chosen, but we can have the gray mode, the void mode, and so on, so that you can choose the representation that you find most interesting to present your data. The second type of representation we have available is the one-dimensional representation. Rather than always working on the same visualization field, we will use the Add Visualization button below to create a new visualization area. Then we will switch to the One Dimension tab. We will choose a violin plot representation, choosing a feature that is the same gene as before, CD3E, and request the display. Here, we have a global visualization of the expression of CD3. But thanks to the Colors tab, it is possible to distribute this visualization through a metadata. For example, we will choose the classification by HTO to see how this gene behaves across the different samples. In each of the samples present, we can see the difference in expression of this gene. It is possible to go back to the first tab and choose a histogram representation instead of a violin representation. We can choose the classification of the samples and thus count the number of cells present in each sample in a histogram. Finally, we will study the last type of representation, which is the table representation. We will add a new visualization. You have a total of 3 visualization zones which allow you to observe your data simultaneously through several types of representations that you choose. In the third visualization, we are going to go to the Table tab, and we are going to ask for the number of cells by choosing the HTO classification, then by clicking on the Split by Cluster option of the Leiden algorithm. The table thus represented gives us in row the clusters and in column the samples, the number in each box being the number of cells present in the cluster and in the sample, which makes it possible to quantify exactly the representation of the clusters by sample. In conclusion, interactive exploration allows you to visualize your data in the most appropriate way in order to answer the scientific questions asked. If you want to focus on particular groups of cells, you can either go to the Differential Gene Expression Analysis module or go to the Subproject Definition module to create a new project from a subgroup of cells. These modules are presented in other tutorial videos.